see we are now recording. Not sure what happened there and why I can't record on Blackboard, but oh well. Nothing like a good. You can still see me, and can you still hear me? Let's just double check that before I head off. Can you still hear me okay? Okay, well, if you can hear me, anyway, we'll do it on Screencast-O-Matic, and I will repost again. I don't know why the recording won't start. And something is randomly beeping in my... Can you hear me okay? Oh, my mic's down there. Let's put my mic up there. Does that help with the sound? Or no? Okay. There, we can hear you. Okay, I'm glad you can hear me. Let's get started because technical difficulties are a pain in the butt, but I'm smart enough now that we have a way of recording. Yay, you can see my face. Look, you can even record me. Hello, online learners. Yeah. Hello and welcome to the joy of problems with technology. Good. Let's click this off. I'll keep my face here just because that's cool. Something is beeping in my system. Not sure what it is. Oh well. Okay, let's continue on with a quick review. Some of you... Whoa, we don't want that. Whoa. Oh, it's scanning my system. Stop. Go away. Okay. Okay, most of you did really good at labeling, so we'll leave it at that. That is good. Descending aorta is another one. Inferior vena cava. You got it all, or good enough, so that's good. Page 2. This was good. Yeah, green is the parts of the nephron. Please realize the glomerulus, though, is not... the glomerulus should be pink because that is... that's the blood vessels right here. The part of the nephron which some of the other groups got is this one should be green, and this is the glomerular capsule, also called Bowman's, capital B because it's a person, Bowman's capsule. Okay, now they're into each other. So the glomerular capsule is right here. It's the outside. See how you've got the blood vessels inside and then you have the nephron that's around it. That's the glomerular capsule. Most of this is really well. Proximal and distal. You didn't label the loop of Henle or the nephron loop. Let's add that just so we have it. Okay, just so we have everything complete. Good. It's already there. Oh, it's already there. Okay. Okay. Now, what I want you to realize is the top part, the convoluted tubules in the cortex, and the loop is in the medulla. And that's because the medulla is a different type of tissue, and it's very salty. So it's salty because of that countercurrent, ascending, descending. One is permeable and one's not, kind of situation. So that's done beautifully. This last bit, you were a little confused. One group did really good. Whereas I think it was group 2 that did really good. Or was it group 3? Yeah, group 2. So you've got 3 processes involved in the process of urine. The first one is filtration. And in filtration, that happens when everything from the blood in the glomerulus goes into the nephron. Everything except blood cells, or red blood cells, and proteins. So everything gets pushed through, and it's pushed through based on what's called hydrostatic pressure, which is the pressure due to blood pressure. So it's very important that you have good blood pressure in order to control your filtration rate. Good. Then the second part is reabsorption, and reabsorption occurs in the proximal tubule right here. So that's good news. And this is where 90% of everything gets reabsorbed. And yes, this is confusing. So I'm cutting in and out. Oh, that's not fun. Okay. Oh, every time I get crazy with the pointer, it cuts out. Okay, we're not getting crazy with, yeah, and my bandwidth is low, so I won't get crazy with the pointer. So you can hear me okay now? Yeah, I'm only down to 1 bar and I've turned red. Okay, so I won't play with the pointer, but it's so fun to play with a pointer. But it's important you hear me. And I also have another program, the recorder, running. So the absorption is in the proximal tubule. And please realize even glucose is reabsorbed there. So we talked about how there should be no glucose in the urine, and that's because glucose gets reabsorbed at this point. So glucose is a small molecule, so it would be pushed through in filtration. But then it is reabsorbed, and 100% of glucose should be reabsorbed in the proximal tubule. And the way it works is sodium is pumped, actively pumped. So there's a lot of mitochondria found in this area. Sodium is actively pumped. That will it'll move a positive. So as a result, negatives will move with it. Then, because you now have a concentration gradient of solutes, water will follow. Okay, you should be able to hear me good now. I'm back to all bars. I'm green again. Yay. Okay. So realize there is a time where filtration, or where things that shouldn't be in the filtrate are in the filtrate. So if there's a problem with reabsorption, you will get all sorts of things in your urine that you shouldn't have in your urine. Okay. So if you test your urine and there's all these extra things in it, that could also be a problem with reabsorption. Good. Then secretion is in the end part, and secretion is finely balancing, where you're taking extra stuff out, you're putting extra stuff from the blood back in. And this is where drugs, and this is where the pH can be balanced. Most drugs are too large to leave in the glomerulus, so they will come back later. Well, now that I've got all that back, I'm just going to see if I can record here properly. No. Oh well. Not sure what's going on there. It's okay, we're recording. Okay, so that is the glomerular filtration, reabsorption, and secretion. Then we talked about in the loop of Henle, this is where you have fine tuning of the water. And the fine tuning of the water is from the fact that the descending, the one going down, can pump out water, and the ascending the water doesn't get pumped. Water moves by osmosis, but the one going up, the ascending, pumps out sodium. And then we talked about 2 different things that control the amount of water. So that's where I left off, I think. So let's close that down. And that should, okay, now I'm good. So let's go back to here. We did that. Another review, we talked about that. We talked about the blood vessels. So I kept my summaries up here. And we made a really good summary of the processes here. So we'll keep with that. Now, we need to come back and talk about the juxtaglomerular apparatus, but I want to talk about 2 things that control the water. So when we talked about the descending loop of Henle, it is impermeable, permeable to ascending. Whoops, let's talk about descending first, because you go down before you go up. We go descending is the down, and ascending. Okay, thank you. Descending is permeable. When you go up, it is permeable. No. Permeable. Water moves as you go down. Water cannot move as you go up. Oops. Sorry, there's so many other little... There we go. Okay. Okay. So, descending, water... Descending. Oh, I took mine away. Where's my Leap of Henley picture? I think I cut it out. Yes, I did. Descending is impermeable to water. Impermeable to water. No. Permeable to water. So water will move if there is a concentration gradient of salt. Realize salt is Na positive, small a. Okay? Then the ascending goes up, you pump out Na plus, depending on the amount of aldosterone. Okay, remember how we talked about aldosterone is a hormone that is produced from the juxtaglomerular apparatus. So let's pop back to the juxtaglomerular apparatus. The juxtaglomerular apparatus is right beside the glomerulus. If there is low blood pressure, it will release the renin. And once it releases renin, that will cause what's called an ACE reaction. The renin will then cause angiotensin to be released and an enzyme reaction which occurs to angiotensin O2, and you can have vasoconstriction. Then that acts on the adrenal glands to produce aldosterone, and aldosterone stimulates the uptake of sodium. So sodium is pumped out. Because sodium is pumped out, water will follow. Okay, so that is one way that aldosterone can affect the loop of Henle in a really long-about way. The other way has to do with water channels. Water channels are affected, aquaporins they're called. Aquaporins are affected by antidiuretic hormone. Now, I asked you to watch the video on how caffeine and urine affect antidiuretic hormone, and they affect it as antidiuretic blockers. So, if you more ADH, well, more water moved back into the blood. So caffeine and alcohol caffeine and alcohol will inhibit ADH. So because while you're drinking caffeine and alcohol, you pee a lot, especially like beer, pee a lot, caffeine, pee a lot. So when you're inhibiting ADH, you will increase urination. That sounds better than pee a lot. If you increase your urination, you will then end up with low blood volume because you're peeing out more than you should. Then if you're not drinking water in between in the morning, when you have a hangover, you have low You have concentrated blood. And remember that word we called osmolarity? That means it has high osmolarity, so it's concentrated. That will trigger your thirst center. But it also will trigger your pain receptors, and that's what gives you the hangover in the morning, okay? And if you notice, if you drink a lot of caffeine and a lot of alcohol, initially you pee a lot, and then the next morning, if you don't drink water, your urine is very concentrated and yellow. That's because it is trying to save the amount of water back in your body. Okay, so on the textbook, it talked about ADH as a damn hangover, is how I relate that ADH is related to aquaporins, and aquaporins is related to peeing a lot and then having a damn hangover. Okay, I'm a big caffeine drinker, so you think you're drinking caffeine to get fluid into your body, but you're not, because your body cannot absorb that fluid. I'm drinking tea tonight, which is a different type of caffeine. Okay, so there are two ways to regulate water that I want you to be aware of. One is aldosterone. Aldosterone is detected by the glomerular apparatus. It comes from the adrenal glands. It increases sodium movement. Water follows. Then the second one is antidiuretic hormone. It comes from the brain, the hypothalamus, and hypothalamus is where the thirst center is. So if your blood is very concentrated, it will release antidiuretic hormone, and antidiuretic hormone will then cause more aquaporins, which result in more water absorption. Okay? Simple. Not very confusing. And for sure, I'll give you a question and make sure you know the difference between the action of aldosterone and the action of antidiuretic hormone. Okay, let's go back to a table here that we haven't filled out yet with the juxtaglomerular apparatus. So the location of the glomerular apparatus. Now, the location is beside the blood vessels of the glomerulus. Okay, so if the juxtaglomerular apparatus will detect three things. So let's look here. Decreased osmolarity. So if you have low osmolarity, remember that osmolarity has to do with concentration. Low osmolarity, what will be the response? Low osmolarity, what will be the response? Think about it logically before we look it up on the textbook. Low osmolarity? Less water, yes. So low osmolarity means your water is very dilute. So you have a lot of water in your blood. So if you have a lot of water in your blood, you want to get that water from your blood into your urine and pee it out. So you have less concentration in your blood. So your juxtaglomerular apparatus is not going to, it's basically going to shut down the process because it doesn't want to. Okay, juxtaglomerular apparatus does not affect ADH. Are we talking ADH here? Okay, good. Increased osmolarity. Oh, keep going. You're doing good. More water in blood. Let's not, you got to be detailed here as in more water in urine or more water in blood, because they're opposite, which is what makes this so confusing. Is you gotta, if you don't follow all the details, you just have a lot of things moving, and then you pee and then you don't pee. Okay, you're doing good. So what's happening to the rest? We've got the condition. And we need to know how it's conditioned. Okay, you're good. Work it through. The Dumbo octopus. Wow, there's all sorts of crazy people. Okay, how are we doing with our summary? Good. Low blood pressure is the juxtaglomerular apparatus. Keep going. You're doing well. I want you to work it through. The last one, low blood pressure. That's a juxtaglomerular apparatus. Keep filling it out. Pull out information that you know. That's the good thing about a summary table, is you can summarize in it. Good. This actually comes right from something somewhere, and I want to find it. Okay, anytime you're talking about renin, you need to finish the rest of the cycle. Renin ends up in aldosterone. Aldosterone increases aquaporin. So let's add another layer there just so you keep it all together. Okay? Okay, ADH is not out. Yeah, no, ADH is yeah, aquaporins, renin, aldosterone, moving sodium. Good. Okay. Good. Oh, okay, we're almost halfway done, and I guess we needed to review this. This shows me that we needed to review, is what this shows me. Okay. Good. We talked about that. We talked about that. We talked about that. We talked about secretion. One thing I want to look at is examples when we talk about reabsorption, tubular reabsorption. Let's put my title where it belongs here. There we go. When we talk about tubular reabsorption, there are things that are actively transported and substances that are passively transported. Which goes in which way? And there's one main one that we're talking about. Okay. The main one pumped is sodium ions. Very good. And then by the process of osmosis, because the sodium moves to create the concentration gradient, chloride moves. And after chlorine moving, we end up with water moving. So a lot of things move based on sodium being pumped. Okay. Good. We've got the important parts there. We just need to talk about hormones and we're done. And even then, we don't need to know the detail. We just need to know what hormones come from the kidneys. Okay, so let's look at hormones that come from the kidneys. First one, vitamin D synthesis is very important. Why is it necessary for vitamin D to be used? Vitamin D will help trigger the kidney to adjust the amount of calcium. So vitamin D is actually related to calcium absorption. And you may realize if you do take calcium substances, you can get vitamin D, and vitamin D and calcium together, which is good. Okay, that's what we need to know there. Urethropoiesis is created by the chemical erythropoietin. EPO, erythropoietin. Erythropoietin is the hormone secreted by the kidneys, which will result in the increase in red blood cells. Did I tell you my whole Lance Armstrong used to be my hero and now he's a big cheater, pumping of erythropoietin blood doping story? No. No one's responding, which means you're ignoring me. Yes, I did tell you about... Yes, okay, so we won't talk about Lance Armstrong injecting a natural substance, erythropoietin, but... Okay, we're done then. We'll talk a little bit more about pH regulation when we talk about blood fluids in the next bit. So I'm ready to go on to chapter 16 now. Sorry, 26 now. Let's take a bit of a break for 10 minutes and we will talk about it. I don't want you to memorize this diagram, but I gave it to you just so you realize there's water in your body. So let's take a break until 10 after 4, and then we will finish chapter 26. I didn't realize how much we needed the review of chapter 25, but I think we really needed a review of chapter 25. So I'm going to pause the recording and keep going from there. Okay, I know I'm starting earlier here, but some people had a question about how... Oh, I'm in the wrong chapter. That's not good at all. Okay, so what the question is how does the juxtaglomerular apparatus affect the distal collecting duct? So the juxtaglomerular apparatus is in this part of the nephron. Juxtaglomerular apparatus will detect blood pressure. If the blood pressure is low, it will send a hormone, renin, to the liver, which activates angiotensin 1, which activates angiotensin 2, which then goes to the adrenal gland and activates aldosterone. And aldosterone will pump out sodium, and in the convoluted, the ascending loop, the one going up, which then creates a osmosis gradient, so water can move based on the gradient. Confusing, huh? Very complicated. And that's how it works. Does that make sense? I'm not sure who had the question. Yeah. So this is a good example of how something in one area will affect another. And if you take any diuretics or water pills, as they're called, interfere with that whole process of angiotensin to angiotensin 2 to angiotensin 1, and they will then stop or block that process, which blocks the uptake of sodium and blocks that increase of water, which keeps your blood pressure low. Which, if you have high blood pressure, is a good thing. Okay. Confusing. The kidney, so fascinating, but so confusing. Okay. And the layers are very important. And if you are interested in the tissues, you can actually read about how the tissues in all the different structures are slightly different too. Good. That any questions on the kidney before we go on to water balance, which looks really confusing, but really it's not as confusing. So if we look at our learning outcomes, oops, they're at the beginning. The learning outcomes for chapter 26 are describe water balance, the components, water balance, and regulation. It sounds like the regulation of water is really tricky, but basically the majority we drink, the majority we pee, and that's in and out. Then talking about balancing, buffers is a little tricky, and the rest is a little tricky. So I'm going to work from a PowerPoint on this situation, so that way I can draw on it, because we all know Jamboards hate me. Well, they don't hate me, they hate my bandwidth. So I included this diagram just so you realize there's water all over your body. A large part of your brain is water, or 80% of your brain is water. About 60 to 75% of your body is water. So everything has water in it. What I thought was interesting is even your teeth have water in it, which to me is fascinating. Okay. Okay, we're still recording. That's good. Good. So this is all the different areas that have water. Now, when we look at the water, there's different names of it. So I'm going to do some drawings. So this way, as we go through it slowly, it will make a little more sense. I don't want to draw in red. I want a prettier color here. So we have cells. Cells to me say, well, so you have cells. This is a cell. Here's a nuclei. Here's another cell. Here's a nuclei. Inside cells is a type of water called intracellular fluid. And that is the water inside the cells. Intracellular fluid. And the majority of the water in our body is inside the cells. Now, change color here. There is actually water outside these cells, and that is called extracellular fluid because it's outside the cells. The extracellular fluid has two different categories. The main one is, or not the main one, the obvious one is in your blood. If you remember, in your blood is plasma. So extracellular fluid is made up of plasma. Then we've talked about this fluid that leaves the blood cells or the blood vessels and washes over the cells, and that is interstitial fluid. Inter meaning between, stitial, which sounds like tissue. Okay. So interstitial fluid is the second type of extracellular fluid. So cellular fluid is divided into two groups: intracellular inside, extracellular outside. Then the stuff outside is divided into interstitial fluid, which is the fluid around the cells, plasma, and a third one, which in this textbook is called other. And other is other locations where there is fluid. So this is lymph. Inside your joints, you have synovial fluid. Inside your eyeballs, you've got vitreous fluid, basically other. Okay, so it's the other location. Now, water can move between these by osmosis, and it goes depending on what you need. So if you have drank a lot of water, we need a different color. Okay, if you drank a lot of water, if you've drank a lot of water, you have a lot of water now in your blood plasma. Excuse me. So that water will move out of your plasma into your cells because there is less or more water in the plasma, less in the cells. So it will move out of the plasma into the interstitial fluid because it moves by osmosis, and then it will also move into your cells if your cells need it, because it's using water for whatever it uses water for. Oops, that arrow's kind of ugly. But it goes into your cells. So you see how water can move from location to location. If you haven't been drinking, a lot of water, your fluid will leave, your plasma will be low. So with the plasma low, it will move out, and the water will leave your tissue, go into your blood plasma, and that will result in your tissues being very dehydrated. Okay, so this is always really confusing when you look at something like this, a pie chart. You've got your intracellular fluid and your extracellular fluid. Your intracellular fluid is the fluid inside. Oh, I've got the wrong word. Intracellular. I called it inter. Let's go back here. Oh, I used this. I didn't label it. So it's intracellular fluid, meaning the fluid inside. Okay, so that is where your fluid is found. Intra and extra are the categories, and then extra is broken down into interstitial fluid, which is the fluid inside your tissues, your plasma, which is the fluid in your blood vessels, and other, which is just your strange compartments such as your joints and your eyes and your lymph and other places there's fluid. Okay, so that should help a little. Now, this is a nice confusing graph, isn't it? Interstitial, oh, it just calls interstitial fluid IF. Sorry, let's make another correction. There we go. Interstitial fluid is IF. This is confusing, but it's basically where are the different components. Let's look at interstitial fluid. Intracellular fluid. Let's start with that. Inside the cell is the beiges. So what's high? Potassium is high. And phosphate is high. Okay. And you've got proteins, which is high, which is important because those are needed inside the cell. Now, what is interesting is outside the cell is blue. Sodium is high. Remember how we talked about sodium and potassium being linked together? So if we go back, here we have our sodium-potassium pumps. So sodium is being kicked out or pumped out, every time potassium is being pumped in. So this is why you end up with high potassium inside and high sodium outside. Then you also need a negative inside, so phosphate goes with the negative, so that way it's not too badly charged. Okay, outside we have sodium. What else do we have outside? Interstitial fluid, chloride, which makes sense because sodium and chloride move together. Oops, right here. Okay, so those are in the interstitial fluid. Sodium is pumped out, chloride follows because a positive needs a negative, so it's not polar. Good. Then, if we're looking at red stuff, what's high in the plasma? Sodium's high, chloride's high, bicarbonate's high, because bicarbonate is and protein is high. Very important because that's in the blood. So let's go back and we have, oops, that's ugly. Sodium is high. Potassium is high, and that's because potassium is a buffer, which we'll talk about. And then protein is high. Okay. So that hopefully makes it a little easier to understand, and then you're not just memorizing stuff. Good. Oh, good, and my sound is still working. Good. So that's basically what this diagram's showing, but I did scribbles instead. Now, when we look at back to the sodium-potassium pump, which we've been talking about for, a good long time now, sodium and potassium are pumped together. So when sodium is pumped out, potassium is pumped in the opposite direction. So that's why sodium and potassium are often in opposite directions in different places. So in order to move things, you move potassium, you move sodium, potassium kind of follows based on the charge of it. Now, when we're talking about blood moving, we talked about already in capillary exchange, in the capillary unit, that filtration is pushing things out, and filtration is due to the blood pressure. So on the end of the heart, or end of the capillary closest to the heart, you have pushing, and the pushing will push it out. Then, so that's called hydrostatic pressure. Then as the blood pressure drops through the capillary bed, the colloid pressure, and the colloid pressure is set up by proteins. Remember that these proteins do not move into the blood because they're out of the blood and into the filtrate because they're too high, too big. Okay? So when we're talking about the movement, filtration out is due to hydrostatic pressure or blood pressure, and the movement into the area has to do with the osmolarity due to proteins, and the proteins, that's called blood colloidal pressure. So that's how water moves in between these these sections. It's based on sodium concentration and blood pressure. Now, we've also talked about facilitated diffusion way back when we talked about transport, and that is that some of the substances are carried in on a special carrier. And that special carrier in this situation is the carrier for glucose. So glucose molecules will be pushed out into the filtrate in filtration, but then in the proximal tubule, there are pumps that will pump carriers that will carry it back in. It doesn't take ATP, it just takes specialized cells. Good. Now, this looks complicated, but we already talked about how ADH is controlled from the hypothalamus. The only thing we're mentioning here is thirst receptors or osmoreceptors. Osmoreceptors are receptors in your hypothalamus, and they will detect the amount of water in your blood. If you have a lot of water in your blood, you have low osmolarity. If you have high water in your blood, that's high osmolarity. And basically, this whole flow chart says it's a continuation of the fact that it's detected by the glomerular apparatus, and it involves renin, and angiotensinogen, and aldosterone. It also talks about your thirst center and drinking. Okay. We've talked about antidiuretic hormone. Antidiuretic hormone is the thirst center, which will then be released from the hypothalamus, act on the posterior pituitary. Posterior pituitary will release antidiuretic, which will increase the amount of aquaporins in the tubule of the kidney, and more aquaporins, more water moves. Okay? And that's just a lot of detail to say the exact same thing. What is the effect of diabetic osmoreceptors? You mean osmoreceptors in diabetics? Okay. Think about the fact that in diabetes, your glucose concentration is set off. So if your glucose concentration is higher in your plasma, that means that you've set up that osmosis gradient and you have a lot of particles in your blood. Because you have a lot of particles in your blood, it will keep water in your, it won't pull water back into your blood. It'll keep it in your urine. And then you pee a lot. Does that make sense? Good. Okay. This last little bit basically just says everything we just talked about. So in aldosterone is from the adrenal cortex. Aldosterone will pump sodium. If your sodium is pumped and is pumped in from your kidneys into your blood, water follows. Good. So it looks tricky, but we've already talked about this. And this is basically the same thing. It's just all the details. And you don't need to know all the details yet. But if you look in some of your pharmacology courses, you will look at blockers. Because every time you have an enzyme, you can have a blocker. And if you block your enzymes, you block this whole process. And then you don't have sodium move, you don't have water move, you pee a lot, and you don't get high blood pressure. Good. The pH scale. We talked briefly about the pH scale way back when. But really, I want you to realize the important thing right now in the pH scale is our blood must stay between 7.35 and 7.45. If it varies from there, we're in trouble. But notice food like grapefruit juice, lime juice, that is very acidic. Now, we say our urine is about 6. But remember, the whole idea of urine is to balance the blood, and then you put the extras into the urine. Good. This is really complicated looking, other than I want you to focus on two terminologies or two words. If your pH lowers, you end up with an acid situation in your blood called acidosis. Acidosis. So there is a compensatory mechanism. You need to get rid of it. It's detected in your brain. You increase your breathing rate. Increase your breathing rate will get rid of your carbon dioxide, and you will increase the pH. Good. Then the opposite effect is an alkalosis. It's not a basis. It should almost be called a basilosis, but it's not. If the pH increases and you get more basic, you have a condition called an alkalosis. And an alkalosis will result in wanting to make your blood more acidic. So you will decrease your respiration rate, and that will keep more carbon dioxide in your blood, which will keep more bicarbonate in your blood, and the bicarbonate reacts with the hydrogen, and that results in the change in pH. A little complicated. Try and get the best you can of the general idea without knowing all the detail. And then the rest of the chapter just goes into a lot of detail. Okay, that's the little spiel. Let's look at the notes to make sure I covered everything I wanted to cover. Oh, I know what we didn't do. It'll show up here. So, we talked about fluid, plasma, interstitial, lymph. Good. Someone filled this in nicely. Beautiful. Beautiful. Okay. Active transport of solutes allows from the movement from blank to blank, from lower you filled it in, lower concentration to higher concentration. Very good. What can pass through a cell membrane without any facilitation? That is just gases. Carbon dioxide and water. Oops, capital O. And water. And even then, water uses aquaporins. Okay? Good. All this basically says to regulate water intake. Above all, basically, how is water generated in the body? We drink it. I know. Profound, huh? How does most water leave the water? We pee it out. Sorry, your urine. Let's not be crass. Okay. So that sounds really tricky, but really it's not. And the rest is just a lot of detail on what we're talking about if you're concerned about the detail. Where is antidiuretic hormone produced? We've talked about that. That's the posterior pituitary. Good. Now, it's also called vasopressin because it causes vasodilation. And that's an old word. Two major effects of vasopressin. That is, it constricts arteries. And if you think of the fact that if you constrict an artery, you will increase your blood pressure, and it also increases aquaporins. I just think that's a cool word. Aquaporins. Aquaporins. Okay? And then aquaporins means more water, more channels or more straws, more water. Okay? Vasopressin, you expect more. Are aquaporins always there and just activated, or they're created? There's kind of a debate whether the aquaporins are created or are they activated. Because it says that aquaporins are inserted in the cell membrane, so that means I think what it is, if you look here, you've got aquaporins sort of being stored in a vesicle. When they're activated, the storage of them hits the cell membrane. So they're there, and then aquaporins actually activates them. So it's not like an open shut gate, but it's more like a an installing is kind of better. It installs it. Does that make sense? Yeah, it's a good question. And this aquaporin is actually something new. Electrolyte balance. Okay, let's go. So it's not like a substitute. It's kind of like an activation. So ADH will, they're sort of, the aquaporin is being stored in the vesicle, and then it actually installs them. ADH causes them to be installed. Does that make sense? Just a second, I need to double check that I'm still recording here. Cool. Takes more than a little technology to shut me down. Okay, this table is a little confusing, but I want you to think about it in terms of, initially we talked about it in terms of interstitial fluid, extrastitial fluid. So where is sodium found? And then we talked about the uses of sodium. So don't think about in this chapter, but think before when we referred to sodium. So an example is sodium is strong outside the cell. Outside the cell is extracellular fluid, which is interstitial, and plasma. Okay? Good. What is the uses for sodium? Where before have we talked about sodium? Action potentials, very good. Muscles, very good. Now, think about the fact that if you're not having enough sodium, it will affect your nerves and muscles. And a very good example of that is heat stroke. If you have heat stroke, you have evaporated too much sodium, and you actually get the jitters and your muscles get weak. So that will affect your nerves. Now, in the textbook, it talks a lot about hyponatrium. Remember how sodium is Na when we look at it, the chemical symbol? That is natrium, so it's Latin for sodium. So hyper means too much and hypo means too low. I want you to realize that there is a difference, but in this course, you don't need to know the difference. You just need to know that it affects it. So what regulates sodium? We talked about something that actually moves sodium in the kidney. What is that? Starts with an L. Yeah, aldosterone. Very good. Okay. Now, sodium usually acts opposite of potassium. And if any of you are athletes, you'll quite often have a banana if you've done a lot of running around, because bananas are high in potassium. So let's look at it. Potassium is the opposite pump of your sodium. So it is high in the opposite areas, which is inside the cell, and it acts as ice in action potentials and muscles again, and it is also related to aldosterone. Okay. Chloride is found where sodium is, because chloride, Na and Cl usually go together because of the charge of Na and Cl. So same thing, yeah. Extracellular fluid, interstitial fluid, and plasma. And again, it is also used in action potential muscles. And there is actually a disease called cystic fibrosis where water isn't moving because your chloride isn't moving. Okay. Good. Bicarbonate. That's one we didn't talk about. We may have to go back and look at the chart where bicarbonate is. Good. Bicarbonate. Bicarbonate is usually found opposite sodium. Bicarbonate's high in the plasma. Good. Good. And bicarbonate is used to buffer. Good. And we haven't talked about buffers yet. We'll talk about buffers in a bit here. Do we get to buffers? Yeah, we talk about buffers as the last little bit. We're doing... Oh, we got 10 minutes left to talk about buffers. So let's finish this off. Sodium is... Yes. Bicarbonate's not really in the... Bicarbonate's big in the plasma. What does... Let's go back to No, let's go back to the textbook and see what that chart is that is tricky to read. This one? Nope, not that one. This one. So, bicarbonate is highest in the light blue, which is intracellular fluid and plasma. Okay. Oh, so it kind of goes with sodium. Sodium bicarbonate, that makes sense. Okay. Good. And calcium is low everywhere. And what's the last one? Phosphorate? PO4, very high in the intercellular fluid. Good, let's finish that off and then let's talk about buffers. Which are confusing but not confusing. Well, it's kind of confusing. That's confusing. Let's not confuse you with a diagram with a lot of chemistry on it. Good. Remember, when we talk about calcium, what's the use for calcium? Strong bones, strong teeth, muscle contraction. Good. Bone growth. Okay. Okay, let's continue on with buffers. A buffer is a chemical that can be put into the body that will prevent the change of pH. So it is a weak acid and a weak base. Okay. And what happens is if it's the weak acid, anything basic will be activated with the weak acid, and the weak base will pick up anything acidic. And the respiratory buffer is fast, and the kidney one is slow. And there are 4 types of buffers I want you to be aware of. Well, one's a subset of the other. Protein buffers. Remember that proteins are these big, long, complicated molecules that will have hydrophilic and hydrophobic ends and little extra charges on the outside. If there's some end that's slightly negative, it can sort of attract a slightly positive end. So as a result, you will have the slightly positive end being attracted, carrying a hydrogen. So really, protein buffers carry hydrogens is what's gone. Okay? Good. Hemoglobin, remember that hemoglobin is a very large type of protein. So hemoglobin is converted into, it's not actually, the hemoglobin can carry the carbon dioxide attract to it, but with an enzyme, carbon dioxide is converted to carbonate ion. And that carbonate ion will then be the weak base, which allows for buffering. Phosphates. Phosphate are HPO4. It's what phosphate is. 2+. Okay. Good. Phosphates can be both an acid or a base because it can then go and it can go to be, it can pick up an acid or a hydrogen and can become H2PO4. 1 negative. Okay. Good. Then we talked about bicarbonate. Normally you've got bicarbonate in your blood. You don't have carbonic acid because carbonic acid is very unstable. And if you breathe, you will change the carbon dioxide, which change the carbonate. And we've already talked about acidosis and alkalosis. So let's just define acidosis. Is when the small p capital H is lowered and alkalosis is when small p capital H is increased. So you're now getting more basic. Now there are two the last bit's very complicated. It talks about metabolic acidosis and respiratory acidosis. Basically realize that metabolic has to do with kidney issues. And as a result, your pH is changing because of kidney issues. And respiratory is due to lung issues. If you're not getting enough carbon dioxide out of your lungs, your carbon dioxide will build up, your bicarbonate will build up, and you will become acidic. Okay? And that, oh, look at that, 1 minute, 2 minutes to go. Yes, I am amazing. Okay? The textbook goes and makes this last bit really confusing and complicated. I just want you to get the general idea of what's going on. And that is the end of metabolic. Oh, I haven't posted the digestive system reading guide. I'll get on that tomorrow. Okay. Good. And we are now done. I'm going to shut off the recording. Yay, we actually